to the class. Yesterday we were discussing about transposon. So we will just start with the transposon. Then today we are going to start after transposon. We will start the cell cycle because in your syllabus it was mentioned that you have to see the cell cycle and its regulation. So firstly, transposon. Earlier, I told you that transposons, they are also known as why we were on this particular page, like why we were discussing about transposon, because earlier we see that within intergenic sequences there are some dispersed repeat, that tandem repeat. So these dispersed repeat, they are the part of lines and signs. Lines and signs, from where lines and signs come in our genome, with the they just originated with retrotransposons. So that's why we are on this particular page that we had to discuss. We have to discuss about the transposon. Transposons, they are also known as mobile genetic element. They are also known as mobile genetic element. They are also known as jumping gene. They are also known as selfish DNA. They are also known as insertional element. So transposons, there are two types, like DNA transposon and RNA transposon. Screen is visible?
Yes, ma'am.
Okay.
Yes, ma'am.
So DNA transposon and RNA transposon. DNA transposon work on the cut and paste mechanism. They just cut and follow the paste mechanism. And here, copy number never increases. Please mute yourself. Copy number never increases within DNA transposon. Copy number remains same. So how it is cut and paste, we will discuss later on. But firstly, we will see the RNA transposon. They are just follow the, here they follow the cut and paste, here they follow copy and paste. Copy and paste mechanism. And their copy number increases. Within the genome, you will find out that they just increase their copy number. And how they do so, we will discuss in detail. So first of all, we will have a look on the DNA transposon. As I told you, they follow cut and paste mechanism. So how it is possible? Just have a look on this DNA transposon here. Just assume that this is a gene. This is a particular part of genome, and here there is one transposon. Now this particular transposon, it will follow the cut and paste mechanism. So there is a cut at these two locations, and this cutted out DNA or cutted out part of the genome, it will just remove from this particular part of the genome and get inserted to any other site within the genome. It get inserted to any other site. So now, as you can see, it get cutted out from this particular site and just pasted into another chromosome. Just assume that it is chromosome 1 and this one is chromosome 2. So this DNA transposon cut it out and get just attached within the another chromosome. So as you can see that it is following the cut and paste mechanism. Here copy number is not increasing. Here the DNA transposon is same in number. Copy number is not increasing. Am I clear to all?
Yes, ma'am.
Okay. Now if in the we just see the DNA transposon. Now what about RNA transposon? Similarly, just assume this is a genome, and within genome there is one repetitive sequence, that is transposon here. So this is retrotransposon. How it is retrotransposon? Firstly, there will be transcription of this particular transposon site, and what we will get mRNA, that is having 5' to 3' end mRNA. Now this mRNA will show reverse transcription with the enzyme reverse transcriptase. Then what we will get? We will get the complementary DNA. This one is the mRNA, and for this mRNA, we just get the cDNA, that is known as complementary DNA. So after this reverse transcription, this cDNA will come here and there will be what? There will be duplication of this helix of the cDNA, and this cDNA get inserted to the genome. So here they are following the copy and paste mechanism. Here it is copying the DNA and pasting it. So that's why the genome size increases and copy number increases for retrotransposon. So by this particular mechanism, we are finding the dispersed repeat and tandem repeats that are a part of lines and sign within the genome. Is that clear?
Ma'am, can you please repeat? My network there was a problem.
Okay. See, retrotransposon, they follow which particular mechanism? Copy and paste mechanism. Genome size will get increased. So firstly, if this is a transposon within the genome, it get transcribed into a particular mRNA, and then there will be reverse transcription of this particular strand of mRNA. So we will have the cDNA complementary to it, that is complementary DNA. Now this cDNA helix will this cDNA helix will get duplicated, and this particular same retrotransposon will be this, and now it get attached to the genome. So genome size increases, copy number increases. Am I clear?
Yes, ma'am. Thank you.
Okay. So I hope it is clear. DNA transposon and retrotransposon is clear to everyone. This is very important topic. Nowadays in various competitive examination, you will get the questions from transposon. So after this, tandem repeat. Any doubt to anyone? No doubt? Okay. So tandem repeat, they are also known as clustered repeat. They are also known as satellite DNA. So repetitive DNA present in tandem, that is one by one, there will be repetition within the DNA, that is known as tandem repeat. So where you will find the tandem repeat, if this is a particular chromosome, this is the centromere and this one is the telomere. Ending of the chromosome, they are the telomere and this is the centromere. So this telomere ending and the centromere, they are the example of tandem repeat. Tandem repeat mainly present in the centromeric and telomeric sequence of the chromosome. Now, why they are known as satellite DNA? Anyone? Why they are known as satellite DNA? Anyone want to elaborate? Why it is?
Because
Mhm.
they are present on some chromosomes, and because of that presence, we can just know which chromosome is this.
Is that so? Okay. Any other thought about this? Why it is satellite DNA? No? See, tandem repeat, if we are saying tandem repeat, they are rich in 80 rich sequences. That continuous repetition of A and T will be there. So if there is 80 rich sequences, 80 rich sequences will be present in the chromosome, then the chromosome density will decrease. If GC rich sequences present within the DNA, then its density will increase. So what we are doing, just assume that we are taking one particular chromosome and we are doing the fragmentation. After that, we are following the density gradient centrifugation. So what we are getting after centrifugation, that three layers in centrifugation tube, we are getting three layers. One layer is of the lowest layer is of GC rich sequences, because the density of these type of DNA, the fragment that are present in the lower part of the test tube, actually they are rich in GC content. That's why the density is very high. That's why they are present in the lower part of the test tube. And in between, in middle layer, you will find out that 80 equals to GC. The fragment that are having equal amount of AT and GC, you will find out them in the middle part. But at the top of the test tube, you will get the fragment, they are highly rich in the AT rich sequences. So as you can see that this AT rich sequences, they are present at the top. That's why we are using the term satellite DNA, because their band during centrifugation, their bands come at the top. Am I clear?
Yes, ma'am.
Yes.
Okay. Now tandem repeat, it is of two types, that is mini satellite and microsatellite. Anyone? Can you give an example of microsatellite? Any example? You all have heard. See, V and TR. Variable number of tandem repeat, that is an example of microsatellite. Here the size of the base pair will be less than 6. The repetition of the base pair will be less than 6. Base pair will be just there. Their repetition you will see again and again in microsatellite. But if you will see the mini satellite, the repetition size will be 7 base pair to 25 base pair. That's why they are mini satellite and these are micro, smaller than the mini. Clear to all?
Yes, ma'am.
Okay. So with this, we had just discussed the basic terms, terminologies related to the nucleus, the chromosome, the DNA. I think important topics we have covered till. Now we are going to start the cell cycle, because we have to just study the cell cycle regulation. So before discussing about cell cycle regulation, we must aware of the term cell cycle. What is cell cycle? What do you understand by cell cycle?
Regeneration of older cells to newer cells by the following the cycle.
Increase in
Which-
cell number, the cycle that helps in increasing the cell number.
Continuous process of cell repairing and growth.
Also repair.
Okay, so it is cell cycle. It is a series of events which will follow orderly and finally lead to cell division. Like you have heard, do two type of cell division, that is mitosis and meiosis. So in that particular mitosis and meiosis, actually there are different events, they are going on in a sequential manner, and that will finally lead to cell division. So mitosis takes place in which type of cell?
Somatic cells,
Somatic cell. And meiosis?
germ cells.
Germ cells.
Germ
Germ
cells.
Okay. So before discussing about your eukaryotic system, we will just have a look on the bacterial cell cycle. We will discuss some lexicon from bacterial cell cycle. So it was explained by which particular scientist? There is one model related to the bacterial cell cycle. Are you aware of that model? Hmm. That model was explained by Cooper, and that model is I, C, and D. What is this I, C, D? Anybody? Anyone? No one? No one? Okay, I think no one want to.
I think it's initiation, then replication and termination.
Yes, yes, you are correct. These are different phases of bacterial cell cycle or different stages of bacterial cell cycle. That this one is the I phase, this one is the C phase, and this one is the D phase. I phase, that is interval phase. Interval phase of cell cycle. This is particularly preparatory phase. It depends on the nutrient status. If there is nutrient, then they will divide, otherwise not. So this is the interval phase. C is the chromosomal replication phase. Here you will see the duplication. Replication is there. So this is basically the duplication phase. And lastly, the division phase. So bacteria divide in how many minutes?
20.
Yes. Generation time in bacteria is 20 minutes. So, and as you know, there are multiple round of replication takes place in the bacteria because the C phase and D phase of cell cycle, they are overlapping in the bacteria. But I'm not going into detail of this thing. So now going to start. This point is clear. ICD is clear.
Yes, ma'am.
Ma'am, please repeat once.
See, ICD is different phases of cell cycle present in the bacteria. This is the model explained by the Cooper scientist. This is the interval phase. Here the bacteria will
Yeah.
prepare for cell division and it depend on the nutrient status. C is the chromosomal replication phase. Here there will be duplication of the DNA. And lastly, the division phase. C and D phase, they are the overlapping phases of bacterial cell cycle. That's why
Bacterial cycle.
yes, that's why bacteria replicate within or replicate or divide within 20 minutes.
20.
Okay. Now going to start the eukaryotic cell cycle. Eukaryotic cell cycle. So how many phases you have heard for eukaryotic cell cycle? How many phases you have heard?
To karyokinesis and cytokinesis is further divided into 4: prophase, metaphase, anaphase, and telophase.
Before that, some interface is there.
It's like
Yeah,
interface
interface,
G1,
yeah.
S, and interface
G2,
G1,
Hmm.
S, G2, and
G1,
M
S,
phase.
and G.
Yes.
Hmm.
So,
4 phases.
if this is a cell, this one is the M, G1 is the more time taking. Then after that, there will be S, G1, S, and G2. This one is
G2.
M. Yes. So G1, S, and G2, they are the interphase. G1, it will long for the 11 hours. S, basically for 8 hours. And G2 for 4 hours.
4,
Yes. And
4
M
hours.
phase, that will be mitotic phase less than
Mitotic phase.
1 hour.
1 hour. So G1 phase, that is the preparatory phase, here cell organelle duplication you will see. And S phase, that is replication of chromosomal DNA. Histone protein biosynthesis also takes place in the S phase. G2 phase, this is the preparatory phase of mitosis. In this particular G2 phase, there will be microtubule biosynthesis takes place. So all these are interphases. Now, before discussing more about these phases, have you heard about checkpoints?
Yeah,
Yes, ma'am.
between G1 and S and G2 and G,
So
S and G2.
firstly, we will discuss what are checkpoints.
Checkpoints are the point verifies the G1 phase is completely the work which was been done in G1 is correctly synthesized in the cell.
Yes,
For
checkpoints, they
proceeding to
ensure.
next phase, checkpoint is the
Yes, yes.
for proceeding to next phase, checkpoint is necessary for
Hmm. Checkpoints, they ensure the ordered event of the cell cycle. That cell cycle proceed in a orderly manner or not. That there after G1 there must be S, after G2 there must be M, and in M different phases are there. So they just check that cell cycle is going on in orderly manner or not. So which are the different checkpoints? Which are the different checkpoints?
Cyclin A, B, C, D, cyclin.
Firstly, G1 to S checkpoints. When the cell will go from G1 to S, so there is one checkpoint, G1 to S checkpoint, that is very important in yeast, animals, and plant. Here, they will check that all the important phases, all the important event that are occurring in G1, it is completed or not. G1S checkpoints. Then in S, there will be DNA damage checkpoint, that after DNA duplication or replication, is there any damage within the DNA? If not so, then it can move to G2. So G2 to M checkpoint, that it will check the event, it will check that spindle assembly is spindle assembly is properly or not. The spindle that are arranged, they are arranged properly or not, basically in the G2 to M checkpoints. Am I clear? G1S DNA
yes,
damage
ma'am.
checkpoints, and then G2 to M. Clear to all? I hope it is clear.
Yes, ma'am. Yes, ma'am.
Okay.
Yes, ma'am.
Very
Now some regulatory protein in the cell cycle. Which are the regulatory protein in cell cycle?
P53. I guess, not very sure.
P53. Before that, some regulatory protein like CDK.
Cyclin dependent.
Cyclin acid decay.
Yes.
Cyclin dependent kinases.
Cyclin P35.
Cyclin dependent kinases, and then cyclin. Some kinases are there that are dependent on the cyclin. They are known as cyclin dependent kinase. They are having, if you will see, this is what? This is the CDK. This is the CDK. So this CDK activity, it properly dependent on the cyclin. It without cyclin, it is inactive. Inactive in state. So what will happen? Cyclin will come and CDK alone is inactive. But when cyclin will come, it will bind in ATP dependent manner. Then this is the CDK, and there will be attachment of cyclin here. So now this one is the active form of cyclin CDK. So this CDK protein remain present in the cytosol. They are inactive or active. It depend on the cyclin. Now what is cyclin? It is a regulatory protein that regulate the CDK. Their fluctuation you will see. They just fluctuate according to different phases of the cell cycle. They just fluctuate. Cyclin fluctuation shown, cyclin protein always in the synthesis or degradation phase. They just synthesize or degrade according to the different phases of the cell cycle. Am I clear?
Yes, ma'am.
Okay, now I'm providing another link. Join on that, then discuss. We will discuss next point.